The basic structure of DNA is made up of nucleotides, which are four basic molecules. Each molecule consists of a distinct nitrogen base. Each one of the nucleotides contains three things: deoxyribose, one of the four bases, and phosphate. If and when there is no phosphate, the sugar and base, instead of creating a nucleotide, create a nucleoside. DNA consists of four bases: adenine, guanine, cytosine, and thymine. Pyrimidines are the complementary bases of cytosine and thymine. Purines are the complementary bases of adenine and guanine. The process of DNA making protein includes two essential steps, one of the two being transcription and the other being translation. At gene expression, we read transcription and translation together. In transcription, information that is stored within the gene's DNA is turned into RNA. Both DNA and RNA are made from nucleotide bases. The nucleotides are chained together to make DNA or RNA. However, each one contains somewhat different chemical properties. So basically, the mRNA is the messenger of the RNA. It's responsible to send the information needed from the DNA to the ribosome. The second step in how DNA makes protein is translation. The information the mRNA sends to the ribosome contains instructions to create protein. The ribosome then reads the sequence of bases. So every codon represents an amino acid. The tRNA, which is called the transfer RNA, groups one amino acid at a time. Amino acids are the building blocks of protein. This process continuously goes on until a stop codon is encountered. The evolution of a living organism is mainly controlled by genes. So, like a mutation can either change, decrease, or lose an expression of a protein. Changes in the sequence of DNA alters all of the copies of the protein. A mutation can be harmful to an organism. Organism's genotype would be all of the genes of the organism. The function and physical appearance of an organism is a phenotype. To be more specific, a genotype indicates if the organism occupies the mutation in a little amount of genes. A phenotype indicates the functional and physical result of the genotype. An important difference among two living things is either if the cell of the organism carries one or two copies of each of the chromosomes. Haploids have one copy of each of the chromosomes, though diploids have two copies of each chromosome. So most single-celled organisms are haploids, but organisms who are more complex and are multi-celled are diploids. Different forms of genes are specified too as alleles. So it's basically like if the gene is normal or if it's mutant. Diploid living organism carries the same alleles, it would be called homozygous. If they carry different alleles, it's heterozygous. If a mutation is recessive, both of the alleles have to be mutant, so that the mutant phenotype is examined. In order for a mutation that is dominant to be observed, the organism must be heterozygous. Recessive mutation leads to a loss of a function, which inactivates the influence of the gene. A recessive mutation is able to tweak the structure of the protein. It can remove the gene from the chromosome and it can interrupt the gene expression. As a result of that, it changes its function. On the other hand, a dominant mutation can acquire a function. A dominant mutation can also lead to a lack of a function. Sometimes, two replicates of that gene are mandatory for a normal function. If one of those replicates are removed, it would cause a mutant phenotype. Sometimes a mutation, if one allele is able to change the structure in the protein, this disturbs the function of the wild-type protein.